Welcome to the second lecture on normal physiology. This lecture will be about physiology of the central nervous system and its role in regulation of motor activities. So, objectives which we will discuss today will focus on nervous control of bodily functions. We will also talk about structural and functional unit of the CNS, which is the neuron. Then we will have their classifications. We will define neural networks. Also, we will talk about glial cells, nerve centers, their properties and principles of coordination. We will define reflex, draw the reflex arc, we will classify the reflexes, and we will discuss excitation and inhibition in the CNS. And at the bottom of the slide, you can see the boxes. This is the progress bar in which each of the boxes it is assigned to the questions which we are discussing, so that it will be easy for you to know at which point of the lecture we are currently in. So let me begin with homeostasis. Homeostasis, it's a dynamic balance. It's actually a relatively stable equilibrium between interdependent elements. And especially this could be related to physiological processes. And parameters that could be regulated in the body could be like temperature, heart rate, pH, levels of hormones. Why do we need to maintain homeostasis? It's essential for survival, and it's essential for all organs and tissues to function properly. In case if homeostasis is lost, it can lead to disease or even death. And how can we maintain homeostasis is by feedback mechanisms, and primarily that should be negative feedback. So how does this happen? Initially we have organism in the state of homeostasis, but as you understand, we can have some external or internal stimuli which lead to changes. And in such case, homeostasis might be lost, and the body will try to compensate for the lost homeostasis. And here we can have two options. In one case, if compensation fails, it might lead to disease or death. But if compensation is successful, then homeostasis will be re-established. So here you can see this is the first question which we are discussing. Now, what about feedback? Feedback, it's a mechanism, it's a physiological loop or regulation in living body that works to bring the body either towards or away from the normal steady state. And we do have two types of feedback, negative and positive feedback, and each of them begins with the initial stimulus. So let's have a closer look at positive and negative feedbacks. So on your left you can see the example of negative feedback. Feedback which tends to damp down or neutralize or counteract any possible deviation from equilibrium, and in such a way it promotes stability. So as you can see on this picture, there is a normal body temperature, but if body temperature decreases, then such mechanism as shivering is activated to bring the temperature back to the normal state. But the opposite is also true. The body temperature might increase. In such case, sweating will be activated to cool down the body. So you have two opposite deviations, decrease in temperature, increase in temperature. But in both cases, as you can see, the body managed to restore the stability, to restore the homeostasis. So negative feedback, it will reverse the changes which were detected. So initial was increase, the body temperature decreases. Initial was decrease, and the body temperature increases. So this is the negative feedback. We also have positive feedbacks. Positive feedback result enhances or exaggerates the original stimuli, so response is amplified. So what do we have in here? The example which we are going to discuss is about the oxytocin level. So During parturition, when the baby and delivery begins, stretch receptors will be activated in the uterus. It sends the signals to the brain, which will increase the release of oxytocin, which in turn causes contractions of the uterus. And this cycle will keep on going. So the more the stretch, the more the oxytocin comes, the more the stretch, the more oxytocin comes, until the baby is delivered. So here you can have an example of amplification of initial deviation. So the more oxytocin comes. And this is all about the concept of neural control. So let's talk about mechanisms of regulation. We have two types of mechanisms. One is humoral, mostly done by endocrine nervous system, and another one is nervous mechanism. So these two, they are the major systems which are used for the communication in the body. They're responsible for the integration of the stimuli and responses if there are some changes in external or internal environment. And these two both are very important for the coordination of functions. And this coordination happens at cellular level, at tissue level, organ level, or even for the entire organism. And this is how physiological regulation happens. Physiological regulation can be defined as the integrated control mechanism underlying various activities. Again, these activities could be at the cellular level, regulation, at the tissue level, or at the level of the organ or system of organs and entire organism. So why do we have two systems? Let's try to compare the nervous and endocrine system. So the nervous system responds to changes very quickly, while endocrine is rather slow. The effects of nervous system, they're short-lasting, while for the endocrine, it's long-lasting. In case of the nervous system, it's the impulse which travels and the response will be highly localized, while for the endocrine system, hormone secretion is rather systemic. And the magnitude of the nervous system effect, it depends on the frequency of action potentials traveling. While for the endocrine system, it's about the amount of the hormone which was released. So that's how the amplitude is modulated. So here it's a brief summary of the comparison, the differences between endocrine and nervous system. So as you can see, endocrine uses using biological active substances. It's rather slow, it's long-lasting, and effects will be diffused and generalized all over the body. And for the neural, it will be using impulse. It's very rapid, short-lasting, it's well localized, and it will have an exact address. So it acts point to point, as you can see on this cartoon animation. So our today's lecture main focus will be on the neural control. So nervous system exerts point-to-point control through nerves, and nervous control, it is electrical in nature, and remember, it's fast but short-lasting. And main function is to control and to coordinate functions throughout the body so that it can respond to the internal or external stimuli. So let me remind you the structure of the nervous system. So what do we have? Central and peripheral nervous system. Central is further subdivided into brain and spinal cord. Peripheral, here we have somatic and autonomic nervous system. In case of somatic nervous system, it's responsible for the voluntary movements in skeletal muscles. And autonomic nervous system, it controls functions of organs and smooth muscles. And CNS is further subdivided into sympathetic and parasympathetic nervous system. And as you might know, in nerve system, you have two types of cells. They are neurons and glial cells. Neurons, they do carry electrical messages called impulses throughout the body. So the structural and functional unit of the central nervous system is the neuron. And here you can see the structure of the neuron. So let's take a look at the functions. We have dendrites, which will receive the stimuli, and soma, which is responsible for the integration of those various inputs. Then the action potential will be generated, a nerve impulse, at the axon hillock. That's the initial segment from where the axon begins. So the action potential will be traveling down the axon towards the nerve terminal. This is where the axon and neuron synapse with the target cell, and that's the place where synaptic transmission occurs. So now, as you can see, we are discussing the second question. Classification of neurons. As you can see, there are many classifications, so let's discuss them one by one. Function. Based on function, neurons are divided into sensory, motor neuron, and intraneurons. So you can see the cell body of the sensory neuron. It's present at the dorsal root ganglion. Then intraneuron is present within the spinal cord at the dorsal horns. And motor neurons, they are present at ventral or anterior horns. Next classification is based upon the structure. In this case, it's based on the number of processes. So you can see there could be unipolar, single process, or pseudo-unipolar. Here one leaves and then it is divided into two. Then there could be bipolar, two processes, and multipolar. Based on shape, you can see based on varieties of shapes of dendrites and somatic morphologies, cells could be divided into many types. So here just few examples like pyramidal neuron, Purkinje cell, also you have basket cells, stellate cells, and so many more other neurons. Based on effects, neurons could be divided into excitatory and inhibitory neurons. This classification, or the effect of that neuron is producing, it is based on the neurotransmitter which will be released from the presynaptic area. And depending on the neurotransmitter on postsynaptic membrane, specific ion channels will be opened. In case of excitatory neurotransmitter, it's about the sodium, so there will be influx of positively charged sodium ions, which decreases the negativity inside, and this is what we call depolarization. In case of inhibitory synapse, the neurotransmitter like GABA, gamma-aminobutyric acid, it is released to initiate influx of chloride ions, and when you have influx of negative ions, it makes the cell, the inside of the cell, more negative. This is what we call hyperpolarization. And in this case, it leads finally to inhibition. Based on axon length, this is one more classification which you might be familiar with. We have Golgi type 1 neuron and Golgi type 2 neuron. Now, based on the neurotransmitter which is released within the synapse from the presynaptic part into the cleft, you can see here there are many different neurotransmitters, and based on that, synapses could be cholinergic, dopaminergic, GABAergic, glycinergic, and so many other examples of neurotransmitters. Now let's talk about glial cells. Here on the picture you can see different types of glial cells, and based on their location, some of them are present only within the CNS and some of them are present in peripheral nervous system. And here you have a summary on main functions of the glial cells. As you might know, glial cells, they are the most numerous cells, even greater in number than the neurons. And some of the functions include, they help to maintain the ionic environment around the nerve cells. They help to modulate the rate of action potential generated and then further propagated. Then they modulate the synaptic transmission because they are responsible for control of reuptake of the neurotransmitters. Then they are also responsible for myelination and recovery from the neural injury. So let's discuss one by one. astrocytes. Astrocytes, these are the cells which are responsible for the blood-brain barrier. So basically they create a barrier between the capillary and the nerve tissue, so that there is no direct contact between these two. They also provide the mechanical support, metabolic function, because they transport nutrients and wastes. Then they regulate the chemical environment, they can absorb the excess of ions, and they can form the scar tissue. Now, microglia, because they're highly dynamic, they can move around, and they act as phagocytes. So in other words, this is a part of the brain's immune system responsible for the defense. Oligodendrocytes and Schwann cells, these are the cells responsible for the myelination. Oligodendrocytes, they do the myelination within the central nervous system, and Schwann cells, they create myelin sheath within the peripheral nervous system. This is how it looks like, with nodes of Ranvier in between. Now, satellite cells and epididymal cells. For the satellite cells, they will form a functional perineuronal sheath around the sensory ganglion neurons at the periphery, and epididymal cells, they are responsible for the cerebrospinal fluid homeostasis, and they play a very important role in it. Now, question number 3, it's about the nerve centers and neuronal chains. So, as you understand, a single neuron will not do much. So, we have groups of neurons, and if this group, it has same function, then this is what we call nerve center. So nervous center, you can say that's a functional association of neurons. So this is a group of closely connected nerve cells which will perform a specific function. And some of such centers, I guess you already heard when you studied anatomy, maybe. We have visual center, we have somatosensory area, auditory center, we have speech centers, we have centers for smell, then we have centers for pain, hunger, and so on. And each of these centers will have a specific location. It could be within the brain cortex or within the brainstem, like respiratory centers, for example. So, nervous center, functional association of neurons. But beside that, neurons, they could be simply connected with each other, like a random chain or random connection. This is what we call neuronal chains. When they are arranged in the form of a chain, that's just a variety of connections in between. And these nervous centers, they might have some properties. So the first property which we will talk about, summation. Summation of excitation could be temporal and spatial. So basically, what happens in such case? You can see there could be various types of inputs, and postsynaptic cell will integrate all of those excitatory inputs, and when a sum of those inputs bring the membrane potential very close to threshold, then it fires an action potential. So summation can change the incoming inputs into the action potential if you will manage to achieve threshold. And we have two types of summation. Spatial summation, that's summation in space, when you have several excitatory inputs and impulses arrive through them simultaneously, and together they will produce greater depolarization to bring it to threshold to have an action potential. While temporal summation, that's summation in time, the other name is consecutive. This is when you have several excitatory impulses coming one by one within a short interval of time. And in such case, postsynaptic depolarization will overlap, like in stepwise fashion, and eventually will also reach threshold to have an action potential. So this is how through summation, even with subthreshold impulses, you can still have an action potential. Next two properties, delayed conduction and one-way conduction. Both are related to the structure of the synapse. So what do we have in such case? As you know, neurotransmitter could be released only from the presynaptic area. This is where it's being stored within the vesicles. So when vesicles get activated by calcium ions, they will start releasing the neurotransmitter so that it can bind with the receptors on postsynaptic membrane. So it could be only one way, because transmitters you have in presynaptic area and receptors are present on postsynaptic area. That's the reason why it's one way. And as you remember, synaptic transmission in chemical synapse, it has many steps, so it takes time. That's the reason for the synaptic delay. The next one, occlusion or blockage. So how does it happen? If, for example, you will stimulate one nerve center, so these circles, they denote the nerve centers with neurons in them. So this is one nerve center with a specific function, and this is another one which also has a specific function. So stimulation of this center activates these neurons, and stimulation of second center activates these many neurons. But if you're going to stimulate these two nerve centers together, you can see the number of neurons which is activated less than you are expecting, because these shared neurons by both centers, they will be inhibited, they will be blocked. So this is the definition of the occlusion. Now the next two properties, transformation of rhythm and after action or prolongation. So in this case, what do we have? The incoming frequency not necessarily will match the outgoing one. As you can see, like here you have 20 Hertz, but the nerve center will receive all the inputs, will analyze them, and will produce the response maybe with the frequency of 70 Hertz, for example. So transformation of rhythm might occur. Now, after action, as you can see, even if you apply the stimuli once, but because of the side chain present in the nervous center, the impulse might keep on circulating within the chain, producing response way longer than you actually stimulated it. Now the next one, fatigue of the nerve centers. Fatigue, that's the feeling of tiredness when you feel like you lack energy or exhausted. So basically it limits you in your performance. So fatigue, it's a brain-derived emotion which actually will regulate how much of work you can do, and thus it helps to maintain the body homeostasis so that you will not get exhausted. And beside central fatigue, which will happen in nerve centers, also we can have peripheral fatigue, which can be developed at the neuromuscular junction. And here you can see some of the reasons for the fatigue, like exhaustion of the neurotransmitter, lack of ATP, accumulation of waste products. Next one is plasticity. Plasticity, it describes how neuronal pathways could be reorganized, rearranged. When this happens, there are two options. Under normal conditions, it can happen when you learn new things. That's when transformation might happen, and that's what we call neuroplasticity. Another example is related to the pathologies. That's what is called injury-induced plasticity. In case if some nerve centers might get damaged, the surrounding cells and tissues surrounding the damaged area, they might undergo some changes in their function and shape so that they can compensate for the lost function. So that's the plasticity. Now, beside properties, we also have some principles of coordination. And here we have divergence. This is when from one neuron the impulse will spread to many. And the opposite one is the convergence, when from many neurons impulses will convey to one single neuron. And example of the convergence is the principle of final common path. So here you can see from various receptors, impulses will come and synapse on one single motor neuron so that similar response will be produced. Next one, it's about reciprocal innervation. Reciprocal innervation, It's related to antagonistic muscles. So how does it happen? For example, if extensor is activated, then flexor has to be inhibited, otherwise no movements will be possible. And that will be happening because of the presence of inhibitory intraneuron within the spinal cord, that's the Renshaw cell. So reciprocal, it's for the innervation of opposing or antagonistic muscles. Now the next one is the principle of dominant. Dominant, that's the focus of stationary excitation and increased activity in the center. These could be described as highly excitable center which attracts excitation from all other areas, which is capable of summation, and this foci is stable and inert. Why do we need this dominant principle? This will help you to choose and focus just on one single thing at a time, because the rest of other needs and wishes will not be taken into account. So you can focus on one thing and complete it. I'll give you one example. If you are hungry, I guess the rest doesn't really matter. So that's the example of dominant principle of how it works. Now, the remaining two, we have feedback principle and subordination. Feedback, actually, we already have discussed. This is what will help us to maintain homeostasis. So we have any changes like stimulus which act on the receptor, impulses will be conveyed to the center, which will make some decisions and will change the activity of effector organs so that they can manage to restore the homeostasis. Now, talking about subordination, this is basically about hierarchy of how nerve centers they are arranged. So of course you know that we do have some superior nerve centers and those which will be subordinated to them. The next question which we have, it's about the reflex. Reflex, that's an action, an automatic response or involuntary response of the organism to a certain situation or stimulus through central nervous system. And here you can see the example of the reflex. When pricking the skin, impulses through the spinal cord will reach the muscle so that it can remove the hand, for example, from the noxious stimuli. And there are two definitions I would like you to know. One is the receptive field. That's a field of a sensory neuron or region or space. If it's being stimulated, it produces activation of that specific neuron and it leads to a particular response. Receptive field. And another thing is the reflex time. Reflex time, that's the time taken from the stimulus until the reaction. Now, what's the structure of the reflex arc? Reflex arc consists of three main components. It has afferent component, center, and efferent component. So afferent or sensory begins with receptor, then goes afferent pathways with sensory neuron at the dorsal root ganglion, which will then enter the center, which could be the spinal cord or brain, and it includes intraneurons. Then at the anterior horns, we will have efferent or motor neuron, which gives rise to the efferent fiber, which will then reach the effector, which could be a muscle or a gland. So basically this is the structure of the reflex arc. Now, there are many different classifications of reflexes. Those classifications could be based on receptors. They could be exteroceptive, proprioceptive, or interoceptive. Then based on centers like spinal, bulbar, mesencephalic, diencephalic, cortical. Then based on effects, reflexes could be motor, motor, secretory, vasomotor. Then classification could be based on biological importance. Reflexes could be divided into defensive, reproductive, nutritive. There is also physiological classification based on the system which is involved, like cardiovascular, respiratory, digestive, excretory, and so on. Also, we could have conditioned and unconditioned reflexes, whether they're acquired or inborn, based on the nervous system, somatic and autonomic, and based on the structure or the number of neurons or synapses, mono and polysynaptic. So in this picture, you can see two types of reflexes. In one case, you have two neurons, like two cell bodies, and only one synapse. So this is what we call monosynaptic reflex. On your right, you can see three neurons. So we have an intraneuron present, and this is what we call polysynaptic reflex, because there is more than one synapse present. Now, let's move on to the next question. It's about excitation and inhibition in central nervous system. So, excitation and inhibition, these are the two main processes which we do have in central nervous system. Excitation, it's about excitatory postsynaptic potentials, and inhibition, it's about inhibitory postsynaptic potentials. So how does this happen? This is example of a cell like resting neuron, and as you can see, the resting membrane potential is equal to minus 65 millivolts. So in such case, if there is an excitatory input which releases an excitatory neurotransmitter, here you can see the examples of excitatory neurotransmitters, which will then open sodium channels and will initiate the influx of sodium ions into the postsynaptic region, it will make the inner surface less negative, bringing it closer to threshold, and this is what we call depolarization. And in such case, postsynaptic potential is being called excitatory postsynaptic potential. So in other words, as you can see, EPSPs, they make it more likely for a neuron to fire, because it comes closer to threshold, and once threshold is achieved, you have an action potential generated. Now, in case of inhibition, there is an inhibitory input, so which means these neurons, they release inhibitory neurotransmitters like gamma-aminobutyric acid or glycine. And when this neurotransmitter binds to the receptors, it will be either potassium channels opening and potassium leaving out of the cell, excuse me, and another option which is available, it's about the chloride influx. So in case if negative ion like chloride will come in, you can see the negativity will become more compared to the resting membrane potential, and this is what we call hyperpolarization, and hyperpolarization brings it further away from threshold, and that's what we call inhibitory postsynaptic potential. And inhibitory postsynaptic potential, this is the reason why action potential cannot be generated. So IPSPs, they make it less likely for the neuron to fire, and this is what happens in case of inhibitory neurons activation with inhibitory neurotransmitters released. So let me remind you the neurotransmission in excitatory synapse. It's pretty much similar to what we have discussed during the neuromuscular junction synaptic transmission in previous lecture. So what will happen is action potential will arrive to the presynaptic terminal, then this will open voltage-gated calcium channels, and influx of calcium into the presynaptic area will stimulate vesicles to travel to the presynaptic membrane to fuse with it, so that the neurotransmitter could be released into the cleft. So once the neurotransmitter is released into the cleft, it then travels to postsynaptic membrane, which has receptors in it, and you have neurotransmitter receptor complex. Once this complex is created, you will have opening of ligand-gated sodium channels. Due to the influx of positively charged sodium ions, there will be depolarization, and that's what we call excitatory postsynaptic potential. And in case of summation of EPSPs, and if threshold is achieved, you can have an action potential generated. After that, transmission has to be terminated. And for that, you have three options. Degradation of the neurotransmitter, or it could be recycled, or it can diffuse back into the presynaptic area. So, for example, if we will take acetylcholine as a neurotransmitter, in synaptic cleft we have such enzyme as cholinesterase, which can destroy the neurotransmitter so that transmission will be terminated. Now, let's have a closer look at some of the steps of synaptic transmission. So, what we have in this case, once calcium level inside the presynaptic area increases, it leads to fusion of vesicles with presynaptic membrane and release of the neurotransmitter. Once the neurotransmitter travels to the postsynaptic membrane with receptors in it, these are so-called ionotropic receptors because they are part of ligand-gated ion channels that have the binding site for the neurotransmitter. And as the neurotransmitter binds to them, ion channel will open so that the ion can travel in or out. And which ion will travel in or out, it depends on the neurotransmitter and the ion channel which is activated. So the nature of postsynaptic potentials will be determined by the characteristics of receptors, means which ion channels are activated and what was the neurotransmitter used. So for example, in here you have activation of sodium channels so that sodium can enter into the cell, and it creates depolarization because you have influx of positive ion. Depolarization brings cell closer to the threshold, and that's what we call excitatory postsynaptic potential. Another two options, they are about potassium and chloride. If it's about inhibitory neurotransmitter, it may open potassium channels and potassium will leave the cell, because let me remind you that we have much more greater concentration of potassium inside the cell rather than in ECF. So that's the reason why you have efflux of potassium, which will cause hyperpolarization since the positive ions they leave, and that's what we call IPSP, inhibitory postsynaptic potential. Another option for the inhibition, it's about the chloride, influx of the negative chloride ions into the cell, which also causes hyperpolarization and leads to IPSP generation and eventually inhibits the transmission. Now, the final result or neuronal firing, whether it will happen or not, it depends on overall sum of the inputs which will arrive. What do we have here? That's a neuron with excitatory and inhibitory synapses, and this is the level of the resting membrane potential, which is -70 for this particular cell. So if it's about the excitatory input, in such case, if input through the excitatory neuron gets activated, you will have depolarization, which is EPSP, and the larger the amplitude of this depolarization, the closer it comes to the threshold, and once threshold is achieved, an action potential is generated. And as you remember from excitable tissues, in the end of action potential, you have after potentials. One of them is hyperpolarization, and in such case, during this period of time, it's more difficult to generate an action potential because hyperpolarization makes it more negative and brings it away from the threshold. And if it's about inhibitory neuron activation, in such case also you have hyperpolarization, IPSP, which is further away from threshold, and again excitation cannot be produced. So in the end, as you understand, each and every cell receives multiple inputs, both excitatory and inhibitory in nature. The postsynaptic cell, the cell body, has to integrate all of these inputs, both excitatory and inhibitory, And when the sum of inputs bring the membrane potential closer to the threshold, it will get fired. And if it brings it further away in the end, no action potential is produced. And along with that, you should know that the redistribution of ions like sodium and potassium, it has to be restored after the firing. And for that you have sodium-potassium pump, which restores the balance. And also you have astrocytes, the glial cells, which will absorb the excess of potassium from the ECF into the cell so that the cell could generate a new action potential. So now let's talk about types of inhibition. We have two types, postsynaptic and presynaptic. Postsynaptic inhibition is pretty much similar to the synaptic transmission in case of excitation. So what happens in this case? Again, same action potential arrives, influx of calcium, which will initiate release of the neurotransmitter, but in this case it's an inhibitory neurotransmitter like gamma-aminobutyric acid, or GABA for short, and this inhibitory neurotransmitter binds to the receptors on postsynaptic membrane so that chloride will go in, creating hyperpolarization, or IPSP. So that's the direct inhibition. The cell is inhibited directly. But beside that, we can have an exoaxonal synapse which does the same thing, but now to the presynaptic neuron. So in such case, this presynaptic terminal will fail to release the sufficient amount of excitatory neurotransmitter, so this membrane will not get excited. So this is what is called presynaptic inhibition or indirect inhibition. So here you have two more diagrams on the same occasion. Like here you have inhibitory synapse and inhibition of this neuron, which is postsynaptic inhibition. And here you have exoaxonal synapse and inhibition of this neuron, which will fail to release enough of excitatory neurotransmitter. So postsynaptic, presynaptic inhibition. Here you have few more examples of inhibition. That's the lateral inhibition through the side chains. The black ones, they are the inhibitory neurons. Then recurrent, when the cell inhibits its own self through the inhibitory neuron. And reciprocal, you remember we already discussed reciprocal innervation. This is when you have antagonistic muscles. For example, if flexors are excited, extensors, they have to be inhibited. Inhibition occurs through the intraneuron. If it's in the spinal cord, then it's about tarential cell. We also have central and peripheral inhibition. They were discovered in experiment. Like in case of central inhibition, the experiment was done on frogs. If a crystal of salt, like normal table salt, is applied to the thalamus of the frog, the withdrawal reflexes of the frog will be suppressed. In case of peripheral inhibition, it happens due to the stimulation of inhibitory peripheral nerve fibers. So that's what initiates inhibition. Now, the second half of our lecture will be about role of different structures in regulation of motor activities. So here we will talk about spinal cord, medulla, then midbrain, brainstem, reticular formation, cerebellum, basal ganglia, and brain cortex, how they contribute to motor control. And as you can see here, you have the new progress bar because we have the new 7 questions to discuss. So let me remind you, this is the overview of the brain structures, and today we will talk about the brainstem functions and how they control the motor activities, and also the cerebral cortex and basal ganglia for the motor control. So motor control, it's about two main things. It's about the posture and movements. As for the postural control, it involves stabilizing a reference. Like you might change the joint angle or redistribute the center of gravity to readjust the position against possible disturbances. Beside that, you can also have postural reflexes, which will help you to maintain the body in upright and balanced position, and also it provides the necessary adjustment to maintain a stable posture. The second one, it's about the movements. Movement control involves reaching a new position throughout the trajectory. Or also, it requires not only the planning, which is about the muscular control, but anticipation of putative consequences of the action. So that's the anticipatory control, so that you can adjust the action and optimize the performance of a movement. So make it well coordinated, smooth, and adjusted. So the first question we will begin with, it's about the spinal cord. You can see that's the first one at the bottom. Let me remind you the structure of the spinal cord. It consists of 31 segments, which will receive information from receptors which are located at the periphery. And these are the sensory inputs. Efferent fibers, they will innervate skeletal muscles. If it's about the somatic nervous system, these are the ventral roots, and visceral organs if it's about the autonomic nervous system. And that brings us to Bell-Magendie's law, which states that the spinal nerves, anterior roots, they consist of motor fibers, while the posterior roots, they contain sensory fibers. And also, if you will take a look at the cross-section of the spinal segment, you will see the gray matter and the white matter. Gray matter, it consists primarily of neuronal cells, their cell bodies, or soma, while white matter is made up of myelinated and unmyelinated nerve fibers. And major nerve pathways which we have, it's about the nerve tracts. They're made from long bundles of myelinated nerve fibers. So here you can see a greater cross-section of the spinal cord segment with all the neurons. But since today our main focus on motor control, we will mostly discuss motor neurons. Those motor neurons which are located in spinal cord, they are called lower motor neuron. And this lower motor neuron is responsible for the transmission of impulses from upper motor neuron to the effector muscle to perform a movement. And we do have two types of motor neurons at the anterior horns or ventral horns. They are the alpha motor neurons, which will innervate the extrafusal fibers, and they are responsible for skeletal muscle contraction. And also we have gamma motor neurons, which will innervate muscle spindles and will control their sensitivity. Besides motor neurons, we also have Renshaw cells. Remember, these are the inhibitory neurons with the glycine as a neurotransmitter. So, functions of the spinal cord, they are subdivided into three: reflex, conduction, and sensory. And our main focus will be the first two, reflex and conduction. So, spinal reflexes are divided into somatic and autonomic. Autonomic will be further subdivided into sympathetic and parasympathetic. Examples include defecation, urination, sweating, and so on. While somatic reflexes will be divided into tonic or postural, which include myotatic or stretch reflexes, and phasic or motor responses, which are further subdivided into flexor-extensor reflexes. Example of flexor reflex, you have protective or withdrawal reflexes. And another subdivision, it's about rhythmic reflexes, those which are rhythmically repeated actions like walking, scratching. And conduction. As for the conduction, you have the nerve pathways or tracts, and here you have the three options. You can have ascending tracts, descending tracts, and also the commissural fibers, which are the transverse tracts. We will mainly focus on ascending and descending tracts. So among ascending tracts, you have dorsal column medial lemniscus system, then you have spinothalamic tract and spinocerebellar tracts. As for the descending, they're subdivided into pyramidal tracts, those which are responsible for the voluntary movements, and here you have corticospinal and corticobulbar tract. And you also have extrapyramidal tracts which control the muscle tone, and they're divided into rubrospinal tract, vestibulospinal tract, reticulospinal, and tectospinal tracts. So here you have example of ascending tracts. Dorsal column medial lemniscal pathway, which controls 2-point discrimination, vibration, and proprioception. These pathways collect impulses from these receptors. Spinothalamic pathway, it receives impulses from receptors about crude touch, pressure, and temperature and pain. So from these receptors to the brain cortex through the spinal cord. And spinocerebellar tracts, they receive impulses from proprioceptors and they deliver this information to the cerebellum. Now here you have the examples of descending tracts. So as we already discussed, they are divided into pyramidal. Example is corticospinal tract, which controls voluntary movements. So it goes from the brain cortex all the way down to the spinal cord. And also you have extrapyramidal tracts, and these tracts will be responsible for the postural control, for the control of muscle tone. And here you have examples of 3 tracts: vestibulospinal, reticulospinal, and tectospinal tracts. Now, let's look at the reflex function of the spinal cord. So let me remind you the structure of the reflex arc. Reflex arc, it has afferent component, center, and efferent component. So as for the afferent, here it also begins with the receptors. Receptors for the reflexes of the spinal cord, they are the proprioceptors mainly. And we have 2 main types, like muscle spindles, or they're also called intrafusal fibers because they're located within the muscle bundle. And muscle spindles, they detect changes in muscle length. Another one is the Golgi tendon organ. These are responsible for the control of muscle tension. From these receptors, impulses will pass to the spinal cord to the motor neurons. And as we already mentioned, there are 2 types of motor neurons. One is the alpha motor neuron, which will innervate the extrafusal fiber, and basically this is the muscle itself, the skeletal muscle, the one which creates the muscle contraction. And another one is gamma motor neuron, which will innervate the intrafusal fiber itself. That's the encapsulated muscle spindle. Why do we need gamma motor neuron with the innervation of muscle spindle? Because we need to adjust the sensitivity of this receptor, muscle spindle, so that it will respond appropriately during muscle contraction. And there is one more thing to be noted. We also do have alpha gamma coactivation. So these 2 motor neurons, they are coactivated so that the muscle spindles, they will remain sensitive to any kind of changes to muscle length during contraction. No matter what, they will still keep on tracking the length of a muscle, and that's possible because of the alpha gamma coactivation. So we have a few examples on the upcoming slides of spinal reflexes. So the first one I want to discuss is the stretch or myotatic reflex. It's about the knee jerk. So this reflex, it's monosynaptic, which means there is only one synapse and there are 2 neurons. So how to produce this reflex? When muscle is being stretched means lengthen. The muscle spindle, which is located inside the muscle, will also get stretched, stimulating the afferent fibers, which will send impulses to alpha motor neuron. And alpha motor neuron, once it gets stimulated, it will cause contraction of the muscle in the muscle which was previously stretched. And as a muscle contracts, it shortens, means it regains its initial length, decreasing the stretching in the spindle and returning it to the original length. So this is how stretch reflex occurs. And at the same time, remember, we have reciprocal innervation. Antagonistic muscle will be inhibited because here you have an inhibitory intraneuron. So, muscle spindle reflexes, they oppose or they're responsible for the correction if muscle length increases or when the muscle will get stretched with the muscle spindle in it. Now, another reflex, spinal reflex, which we have, flexor withdrawal reflex. That's example of the phasic reflex. This reflex is polysynaptic. So here you have more than 2 neurons and we have 2 synapses. And when reflex is initiated, there will be flexion on same side and extension of the opposite arm for the balance. So this reflex, it comes from somatosensory or pain afferent fibers, which will produce withdrawal to remove your arm from the noxious stimuli. In most of the cases, it's about the pain, like touching something hot, like hot object, like burning candle in this case. It stimulates the flexor reflex. And once receptors will get activated, impulses will pass to the spinal cord, to alpha motor neurons to activate flexors so that the hand will be removed from the noxious stimuli. So on same side, ipsilateral side, you have flexion and cross-extension reflex on the opposite side to maintain the balance. So why do we have those reflexes and why do we need to study them? Muscle stretch reflexes or deep tendon or myotatic reflexes, they are involuntary contractions of muscles which will be induced by a specific stimuli to a specific receptive field. So as you can see, you have to hit with the neurological hammer a very specific area so that the reflex will be elicited. And their centers will be located in specific segments. So clinical significance of these kind of reflexes, they can indicate the pathology along a particular nerve route to a section of the spinal cord. So if there are any damages on the way or in the center, or it's continuous and works perfectly. Now, the second question, it's about the medulla and pons functions. So in medulla, you have vital reflex centers. Vital means life-sustaining. Examples include vasomotor center, which controls blood pressure and cardiac activity. Then you also have respiratory center, which controls rate and depth of breathing, along with pons, which has pneumotaxic and apneustic centers for the regulation of respiration. Plus, you have defensive reflexes like vomiting, coughing, sneezing, blinking. Then you have defensive reflexes like digestive reflexes like deglutition or swallowing, mastication or chewing, salivation and sucking reflex in newborn babies. Plus you have the nuclei of the cranial nerves in both medulla and in pons. Now let's take a look at midbrain functions. Midbrain consists of tectum and cerebral peduncles. In tectum you have superior and inferior caliculi, which are the subcortical visual and auditory centers. And here you have orienting reflex. Orienting reflex, that's involuntary response, like turning the head towards the novel environmental stimuli. It could be sound, or it could be a visual stimuli. And the person will pay attention to it even before identifying. So that's the orienting reflex. You tend to turn towards the new stimuli, visual or auditory, to check. Now, cerebral peduncles, here you have tracts. Also you have substantia nigra with dopaminergic neurons, which play a very important role in modulation of motor activities. And also you have tegmentum with red nucleus, that's another important structure. And red nucleus, this is where the rubrospinal tract begins, which will facilitate the muscle tone of flexors, and it's responsible for the inhibition of extensors. Now, the brainstem, which includes the midbrain, pons and medulla, it has two main functions, conduction and reflex as well. And conduction, it's about same ascending and descending pathways. while brainstem motor reflexes could be divided into static and statokinetic. Static reflexes are further subdivided into postural or supporting. Examples include tonic neck or tonic labyrinthine reflexes, and righting. Righting means they help to maintain an upright position of the body. And here you have neck, body, limbs, righting reflexes. Beside that, we also have statokinetic reflexes. They are concerned with both angular, rotatory, and linear movements or accelerations. Now, brainstem, it plays an important role in control of motor activities. How will it happen? Because it exerts facilitatory and inhibitory influences over the spinal extensor reflexes, and they could be divided, as you can see, into two types. Such structures as frontal cortex, basal ganglia, cerebellum, and medullary reticular formation will largely inhibit spinal extensor circuits, while the pontine reticular formation and vestibular nuclei, which is the Dater's nuclei, they will facilitate them. So how can we prove that? We can use sections at different levels, and this is what was done in experiments on animals a long time ago. So for example, if you will make transection of the brainstem above the midbrain, there will be no major changes in the muscle tone. But if you will cut the brainstem right below the red nucleus, which means basically you remove it, you will remove the influence of the red nucleus over the flexors. And this is what is called decerebrate rigidity, because in such case, stimulation of flexors no longer happens, and this animal is left only with the influence of Dater's nuclei or vestibulospinal tract, which, as you have seen, is responsible for facilitation of extensor muscle tone. So this is how the animal will look like. This is called decerebrate rigidity, extensor posturing. It describes the involuntary extensor positioning of limbs and the body, because flexors are no longer being stimulated in such case. And if you will make a transection below the third one, it will cause a flaccid paralysis. Now let's talk about reticular formation. It's a diffuse mass of neurons and nerve fibers within the brainstem, and it can have two types of influences: ascending and descending. Ascending towards the brain cortex, it's about ascending reticular activating system. It plays an important role in arousal, alertness. It helps to maintain an attention, focus. It controls sleep-wakefulness cycle. While descending influences towards the spinal cord, here you have two groups of nuclei, pontine and medullary. As for the pontine, it will exhibit generally a stimulatory effect on both extensors and flexors, with the predominant effect on extensors. While the medullary part, it exerts an inhibitory effect, so it does the opposite. Beside that, reticular formation also helps to control autonomic functions like cardiac, blood pressure, respiration, the GI tract. Now, the next structure which we will talk about is cerebellum and how it controls movement. Cerebellum is divided into three areas. We do have vestibular cerebellum, which is responsible for the control of balance and eye movements. Then we also have spinocerebellum, which is responsible for the synergy of movements, like controlling the rate, the force, the range, the direction of movements. And also we have the pontocerebellum, which will do the planning and initiation of movements. And from histology, you might remember that in cerebellum you have the cortex, which is three-layer structure, And all of the outputs from the cerebellum, they begin from Purkinje cells, and they will be inhibitory in nature, because Purkinje cells, they're inhibitory cells with GABA as a neurotransmitter. Now, functions of the cerebellum, they include coordination of gait. It helps to maintain the posture and equilibrium. Then it controls the muscle tone and it helps to control the voluntary muscle activity. Then it is also responsible for coordination and regulation of well-coordinated muscular activities, skilled movements. And why all of that is possible? Because cerebellum, it has 3 pairs of peduncles which connect the cerebellum with all the structures of the brain, and that's how it exerts control over the spinal neuron. That's how it receives the feedback and all the afferent and efferent inputs. So if cerebellum will get damaged, there will be some symptoms. So some of them are listed in here. So the first one, dysadiadokinesia. This is lack of ability to perform rapidly alternating movements. Ataxia, that's the poor muscle control, especially during walking, like clumsy walking will happen. Nystagmus, uncontrolled rhythmical oscillations of the eyeballs. It could be horizontal or vertical. Intention tremor, it happens when the person tries to perform a voluntary movement. Then dysarthria or scanning speech. That's how the person will talk to others. Hypotonia, that's what is called decreased muscle tone. As the muscles, they lose resistance to palpation due to diminished influence of the cerebellum over the gamma motor neurons. Also, there will be lack of balance and impaired ability to estimate the distance. Another group of structures which is also important to discuss, basal ganglia. Basal ganglia, that's the group of anatomically connected and functionally related nuclei. These are the structures which are included into the basal ganglia, and basal ganglia receive inputs from areas of cerebral cortex, like basically all parts, and they will relate it to the thalamus, and also all the structures within the basal ganglia are interconnected. Basal ganglia will modulate the thalamic outflow to the motor cortex so that you can plan and execute a smooth, fine movement. Basal ganglia also control the muscle tone, and they control the automatic associated movements. Besides, they also control the reflex muscular activities. So here you can see the location of those structures on the cross-section of the brain and how they could be grouped, and these structures are included into the striatum. We will need it when we will discuss the pathologies. So, lesion of the basal ganglia. If striatum will get damaged, it leads to deficiency of GABA as a neurotransmitter. Here on this diagram you can see connections of the basal ganglia to cerebral cortex, to the thalamus, and in between, and which neurotransmitters are released. So in case of striatum damage, deficiency of GABA leads to hyperkinesia and hypotonus. Hyperkinesia, excessive involuntary movements which could not be controlled or reduced. Hypotonus, decreased muscle tone. Then, if substantia nigra is damaged, you remember there we have dopaminergic neurons. It leads to hypokinesia, lack of movements, and hypertonus, increased muscle tone. And here you have examples of lesion of separate structures like globus pallidus, responsible for the postural support, subthalamic nuclei, which will produce wild, uncontrolled movements. Then lesion of the striatum we already discussed, an example of the disease like Huntington disease. And I guess the most well-known and well-studied disease is the Parkinson disease. That's when there is a destruction of dopaminergic neurons, And in case of Parkinson's disease, here you can see how the patients will look like. They will have tremor, they will have rigidity, they will have mask-like face, the trunk will be tilted forward, reduced movements. So that's the Parkinson. It occurs in case if there will be loss of dopaminergic neurons. Now, finally, we will discuss the brain cortex. That's the highest structure which is needed for voluntary control of movements. So the entire brain cortex could be divided into three areas. They are sensory, motor, and association area. Examples of sensory areas, they include primary somatosensory area, auditory area, and visual cortex. Motor areas are further subdivided. They're all located in frontal lobe. And here you have primary motor area, which is in precentral gyrus, then premotor cortex and supplementary motor cortex. For the association, you have prefrontal area, then parieto-temporo-occipital area, and limbic. So, as you remember, the brain cortex, it has a 6-layer structure histologically, and it can be divided into individual columns. Like a column, it consists of neurons from all 6 layers, which are located perpendicular to the surface and that are responsible for one same function. So in the end, you will have lots of cortical columns which do one thing, one function, like if it's one movement, or it's responsible for one particular sensation or modality. And if you will take a look at the structure, histological structure of the brain cortex, you can see that sensory association area they differ from the primary motor cortex, because here the layer where you have the bad cells, it occupies significantly larger space, because from primary motor cortex, that's the place from where descending pathways, corticospinal tract, from where it begins. Now, we also have such thing as Brodmann's areas. Originally, we used to have 52 regions, and all of them, they have different histological structure and cellular organization. I will give you 2 examples of such areas. One is called primary motor area, which is located, that's the area 4. It's located in precentral gyrus, and it's responsible for the execution of voluntary movements. And in this area, if you will take a look at it, projection of various regions of the body can be plotted using motor homunculus. Motor homunculus with muscles of the face located at the bottom of the precentral gyrus, with limbs and body going to the top. And as you can see, face and arm, it occupies significantly larger area rather than trunk and legs. So it's not proportional to the real human body. Beside primary motor area, we also have primary somatosensory area, which is located in postcentral gyrus, that's the area number 1. Here you have such sensations as touch, temperature, vibration, pain, sensory perception like 2-point discrimination, proprioception, and so on. And here you also have sensory homunculus. The same thing. Face and arms, they occupy the larger area because these are the most important parts of the body which we are using the most, while trunk and legs occupies much more less space. Now, our today's main focus will be on motor cortex. So once again, it's divided into 3 regions. Primary motor cortex, that's the precentral gyrus, remember about homunculus, area 4. It initiates voluntary movements, and from here the corticospinal tract and also corticobulbar tract, it begins. Now, premotor cortex, area 6, it generates a plan for you to make a move. Also here you have area 8 for the movements of eyeballs, and supplementary motor area, which basically does the same. It programs complex motor activities. So For you to make a move, any kind of a voluntary movement, you should have the idea of what you want to do. That's about cortical association areas, prefrontal cortex. Then you also require basal ganglia, cerebellum, subcortical structures to make all necessary adjustments, correction. After that, impulses will go back to premotor cortex, premotor and supplementary motor cortex, so that it will make a plan. It will program a sequence of movements. And then finally, primary motor cortex will do the execution. Every time you make a move, a feedback will be sent to the brain cortex so that you can evaluate whether the move you make, if it's fine, well coordinated and smooth, or maybe some adjustments are needed. So here is the summary of regulation of motor activities of the body and the structures which are involved into it. So as I've already mentioned, cerebellum, subcortical structure, it will build an internal models and it will correct all the motor commands which come from the brain cortex, because it's responsible for comparison of what you do to how it should be. Basal ganglia, it provides optimal control about the motion, it can facilitate motor signals. Then you also need the parietal cortex for sensory inputs, because it will receive impulses from proprioceptors, and also it collects like visual cues, auditory cues, vestibular, touch, and so on. And it plays role as an estimator. It integrates all the multimodal information. You also need premotor cortex and motor cortex, which will transform your predictions into sets of motor neuronal discharges. So basically it creates a program of like which neurons in which sequence and for how long they should be activated, encoding for force and direction of movements. So basically these are the summaries of how any movement is being regulated. Thank you for your attention and have a nice day.